Dr. Bradley Voytek is currently a postdoctoral researcher in the Gasley Lab at the University of California, San Francisco. He received his Ph.D. at the University of California, Berkeley, working closely with Dr. Robert Knight, who many of you may remember from the Children's Hospital presentation last year. I guess that was only last semester. Time flies. Brad has participated in a number of really fascinating studies involving novelty processing, brain networks, and the underlying physiology that mediates cognition. He's been involved in developing brain-computer interfaces and has many colorful side projects that I'm sure will be introduced at some point. He's an avid educator, a prolific writer, and his work has been featured in many publications, including Wired, Scientific American, the New York Times, Forbes, and many others. He also keeps an excellent blog, which has been regularly fascinating to me, and I highly recommend you check it out. You can find links to it on our website or his. Tonight, Dr. Voytek will share some of his recent work on the wide-ranging functions of the frontal lobe. There will be time for question and answers afterwards, but since he can't stick around, wonderful. So there will be time for question and answers afterwards, and he will be able to stick around to field those. So that said, it's my great pleasure to introduce Dr. Bradley Voytek.
So first of all, thank you, Berkeley, for having me, and thank you, Science Club. This is cool. So going to try and unpack a little bit of the research that I do. I decided to scuttle some of the more detailed stuff in favor of the cooler stuff. I'm currently a researcher at UCSF, and most of the time I'm working directly with people. I don't do really animal research, and the kind of research that I do is a little bit wide-ranging, and so I'll try and keep it consistent. Basically, I'm curious as to how it is that we go from this, which is clusters of hundreds of millions, or billions, I should say, neurons, to this, right? Or sometimes, sadly, even this. And to approach that, I try and take 4 different but supporting tactics. This is the only slide I have with text on it, by the way. So I look at causal links between brain and cognition. So for a long time in neuroscience, I think many people are familiar with brain imaging studies. And most brain imaging studies are correlational. So they say, what part of the brain is working harder when I do something harder? So if I make you, try and remember a bunch of items, like remember 3-digit numbers versus a 7-digit number, I say, what parts of the brain are more active when you're remembering 7 digits versus 3 digits? And when I see that, I say, okay, those are the brain areas responsible for memory. The analogy I like to use is there's a problem with that, which is if I have you running on a treadmill and I'm recording activity from your arm muscles, let's say, and your leg muscles, then the faster you run, the harder your arm moves. And then if I was a cognitive neuroscientist, I would say, aha, you're moving faster, and your arms move faster when you're running faster, therefore your arms is where running happens. Correlation doesn't always imply causation is basically what I'm getting at, right? And so it's harder to look at causal relationships between brain and behavior in neuroscience. But we can do it, it's just messy. 2, intra and interregional communication. That is, how do different parts of the brain actually communicate? Now, again, most brain imaging studies are saying what parts of the brain are active when you are doing memory. And really, there's a lot of baggage from psychology in neuroscience. Not that it's a bad thing, but for a long time, we didn't really understand neurons. We didn't know that there were neurons up until the early 1900s, right? Late 1800s. And then we didn't really know how neurons communicated or anything like that. And so for a long time, psychology and neuroscience was saying, where in the brain is memory, or where in the brain is attention, or where in the brain is language? And what we should be asking instead is, given what we know about how neurons work, how could neurons work together to give rise to behaviors that look like what we call memory and attention in language? Does that make sense? There's a subtle difference there, but I think it's a very important one. And so one is trying to take a top-down approach, we would call it, which is, where does this behavior happen? The other one is taking a bottom-up approach, as we call it, which is, how do neurons work together to give rise to behaviors? And that's what I think is really important. 3, the plasticity of cognitive networks. Plasticity in this case means changeability. So when we are remembering something, when we learn something new, there's a physical change happening in the brain. And that happens all the time. Your brain is always changing. And we call that plasticity. Your brain is very plastic. And which is infuriating when you see people who write articles that say that the internet is changing our brains, and therefore that's bad, right? Like it's somehow harmful. Everything that you do changes your brain, right? Change is not bad in this case, right? Where change happens or what kind of change happens, that's what's important. And number 4, population-wide analyses of cognition. So we have enough data now that we can start looking at much larger scale questions, and I'll get to that in a little bit. So first of all, what is executive control? This is what I'm interested in. And executive control is a term that we use in neuroscience. This is one of the primary functions of the frontal lobes, or the prefrontal cortex. So just to orient you, very cleverly, at the front of your brain, right above your eyes, sits the frontal lobe. And for a long time, surgeons basically considered the frontal lobe to be mostly useless, especially the prefrontal cortex. And that's because they would go in and do surgery, and they would have to cut out parts of the frontal lobe, and it didn't really look like it was having any severe effects on people's behavior. And so they said, oh, it's not really that important, right? If you damage somebody's motor cortex, or you cut into it, if you're a surgeon and you have to, for whatever reason, remove part of the motor cortex, let's say you have the tumor, or they have a stroke that damages it, then you'll be paralyzed on the opposite side of the body. If you damage the visual cortex in the back of the brain, then you will be cortically blind. You won't be able to see objects. But if you damage the prefrontal cortex, for a long time, people didn't really think that there was much damage. And so that's part of what I work on, is to try and figure out what is all of this at the front of the brain, which we think is very critically important for our cognition. What is it doing, and how does it do it? And this graph, this very complicated looking graph, is from a paper that my wife and I put together, which basically took 3 million peer-reviewed publications, so scientists publish papers to get their information out there, and we took all 3 million papers that have been published on neuroscience and did some data mining on it, meaning we grabbed phrases and saw what papers talked about which related phrases, and from that we can infer how things in the neuroscientific literature are related. And so executive control, which is what I'm interested in, is very closely related to these 3 brain regions, which are all in the frontal lobe. These are subregions of the frontal cortex. So you have the prefrontal cortex and the dorsolateral prefrontal cortex and the anterior cingulate gyrus. And then you have all of these clusters of cognitive functions that it's involved in. There's all of these tasks and things that scientists have people do. And so there's like a trail-making task and a Stroop task and a flanker task and attention and executive functioning and central executive and multitask. And all of these things, these are all part of what we call the central executive or executive control. So to give you an idea of the important role that our memory and experiences and attention play in our perception, I'm going to play a really annoying sound right now, and I apologize. Okay, so who understood what was being said in that? Okay, on average, if I take a big group of people, I think people on average get about 2 words correct out of this. Now I'm going to play an unfiltered version of that. So that's just a sentence that we recorded, and then we filtered it out. I'm going to play an unfiltered version. Hopefully.
She writes to her brother every day.
Okay, how many people understood that? Good. Now I'm going to play the exact same thing I played the first time. How many people understood it? Okay. Did it sound different the second time than it did the first time? Like qualitatively, did it feel different to you? Yeah. It's the exact same stimulus, but now you have a memory that's filling in the missing information, right? And so that is what I find fascinating, that we can use our experiences. In this case, we call that working memory. It's a very brief memory from just a few seconds ago to fill in missing information in our perception in the rest of our world. And to give you an example of the neuroanatomy of this, for every one, so you guys know you have brain cells, neurons and axons, right? Axons are the wires that connect neurons. That's like the main communicating cable. For every one axon that goes from your eye back to the visual part of your brain, right? So for every one piece of information coming into the brain sends 10 axons back onto the visual cortex internally. So for every one piece of information you get from the external world, your brain is filling in 10 times more information. That's not exactly quite accurate, but that's a rough idea of the anatomy of how severe this actually is. So let's talk about causality in neuroscience. How do we get to causal links between brain and brain function? So classic cases here. This is a gentleman known as, his name is Phineas Gage, and he sort of kicked all of this idea that we can maybe link complex behaviors like emotion to the brain. And this rod that he's holding it's about this tall, and it got shot through his head, up through his brain, the frontal cortex, and out, and he managed to survive. This was in the 1860s. He was a foreman at a railroad company, and they used these big iron rods to push basically explosives into holes that they drilled in rocks, and that would let them blow apart big rock faces. And unfortunately, when he was pushing down this explosive, it went off and sent the rod shooting up. And he managed to survive despite the care of his doctor, and which was not fantastic. The doctor tried to get the shards of the guy's skull out because the skull got pushed up in. And so the doctor, because they didn't really know much about the brain at the time, just sort of stuck his fingers in and fished around and ended up damaging a lot of other brain tissue probably. And this is a classic picture from back when lobotomies were being performed regularly. And this is, I believe, the hand belongs to Dr. Walter Freeman, who is a serial lobotomist. He was like the expert, and he would actually just line people up, and he would do this sort of as a display in front of crowds. And he would just go through with an ice pick and go, next person, and just lobotomize like dozens of people in a few minutes, because it was remarkable in how it appeared to cure people. I mean, it won a Nobel Prize because it was a remarkable treatment, but it's sort of like electroconvulsive therapy, which is actually a very good treatment for certain conditions, started becoming overused, and there was a really big public backlash against it. But moral of the story here is that brain damage actually tells us a lot about brain functioning. So back to the person running on the treadmill case, and I think, okay, arms are where running happens. In order to disprove that, all you need is one person without an arm to start running on the treadmill to prove that you can run without an arm, right? Similarly, in neuroscience, all you need is one person that has some kind of specific brain lesion. I could stick 5,000 people in a brain scanner and say, this part of the brain right here lights up every time, have people remember something. And then find one person with a lesion to that part of the brain, and they can still remember something. Then you say, well, okay, maybe that's not exactly where memory happens. That's not where remembering happens. It's just part of the remembering network, right? So this is a reconstruction of the path that the rod took in poor Phineas Gage. And this is actually pictures of brains of the famous case of Paul Pierre Broca's patient Tan. And he was called patient Tan in the literature because he couldn't actually speak. He couldn't generate any kind of words except for the word tan he would say over and over again. Broca saw many of these kinds of people who had what we now call Broca's aphasia, which is the inability to generate speech. And he looked at their brains after they died and found that they all had lesions in generally the same area right here, and usually on the left side of their head. And so Broca said, aha, this part of the brain right here on the left side, that is responsible for generating speech. Kind of an amusing story, actually, if you read the literature. Patient Tan here was called Tan because supposedly all he could say was tan, but if you go back and read Broca's original papers in French, he cursed a lot as well, but referring to him as patient Merde in the literature was not really seen as appropriate, I guess, so they called him Tan. So how do we study things like memory? How do we study things like attention? So I'm going to show you now a classic task that we use to study working memory. Your job is to pay attention. You're going to see 1, 2, or 3 squares flash on the screen, disappear for about a second, and then the same number of squares reappear in the same exact location. And your job as a subject of my study is to tell me, did one of the squares change color or not? And you just press one button if they change color and another button if they didn't. Yes, right? Yes? No? Yeah? No? Okay. Okay, so this is how we study memory. So every time you see some groundbreaking study in the news about we've discovered where working memory happens, mind you, what's happening is there are 20 poor undergraduates from UC Berkeley sitting in a room staring at a screen for 3 hours pressing buttons while this happens. This is memory, right? And this is great because we're really trying to control for all variables, right? You're really trying to distill down everything except for memory, right? But of course, the real world doesn't work this way. It's not boxes and squares, right? Usually, unless you're in one of my studies. But nevertheless, this is what we have to work with. And I have a colleague, a friend of mine at Vogel up at University of Oregon, showed a couple of years ago in a really cool paper that if you record EEG, which is passive recording electrodes, you put a little cap on somebody's head and you record the electrical activity coming off of their brain, you could actually predict a person's working memory capacity based upon their EEG. So this is EEG activity. This is these little squiggly waves, right? This is what we record. This is our signal in neuroscience. And when you remember one item, the signal's not very strong. When you remember 2 items, it gets stronger. When you remember 3 items, it's even stronger. And 4 items, it sort of maxes out because on average, people remember between 3 and 4 items. And this is basically the ability showing each individual dot here is a subject and their working memory capacity by their EEG activity. So you can actually predict a person's memory by their EEG. And this is really cool. But again, that's correlational, right? So we wanted to look at causal relationships. So I worked with a group of 6 patients who had brain damage, very specific focal brain lesions on either the left or right part of their prefrontal cortex. And this image here is the sort of overlapping brain scans. And the more yellow and red it is, that means more people had a lesion in that exact spot. So here, one person in purple had a lesion in all of those regions, and these are slices this way through the brain. And all 6 subjects had a lesion right there in that spot. And then I had another group of 6 patients who had much smaller lesions to a very specific other part of the brain called the basal ganglia. And both of these brain regions we think are very important for working memory. But again, most of the studies are correlational, so we wanted to look at causal relationships. And so when we look at this, what we find, the hypothesis is, and this is really, I'm going to jump to the next, give you a quick primer on how vision works. So everybody, I think, probably knows that if I move the right side of my body, my right arm, it's the left side of the brain that's controlling that. And all the sensory information I get from the right side of my body goes to the left, and all the sensory information from the left side of my body goes to the right. Vision kind of works similarly, but not exactly. So if I'm looking straight ahead, it's not that stuff from my left eye goes to the right and right eye goes to the left part of the brain. It's actually information on the left side of my world goes to the right side of my brain, and information on the right side of my world goes to the left half of my brain. And there's a very basic reason for that, is that the eyes are cups, the retina are cups. And so the outside part of this eye gets information over there, and the outside part of this eye gets information over, the inside part of this eye and the outside. And the information ends up going like that. And that's really important because basically, if I have a brain lesion here, and I present a stimulus to this side of the world, the stimulus enters the good half of the brain, the non-damaged half of the brain. And what it turns out is that if you have a brain lesion to the prefrontal cortex, and I present you a stimulus to the good half of your brain over here, you're behaviorally just fine. You don't have any problem remembering items. This is people's memory accuracy. So control subjects, and then patients with brain lesions. But if I present a stimulus to the bad half of the brain, so a lesion over here, and I present a stimulus over here, then suddenly you have impaired working memory. And so I can actually, using knowledge about neuroanatomy, and basically how information enters into the brain, we can sort of start piecing out apart how the brain interacts. And so what's interesting is that if I look at activity in the back half of the brain, in the visual parts of the brain, there is a decrease in visual activity, or activity in the visual parts of the brain, only on the side of the brain that has lesion. And so what's happening when you see that first group of 3 squares, let's say, flash really quickly, what's actually happening in the brain? When you see that second group of squares flash, what are you comparing that second group of squares to? You don't actually have anything on screen to compare it to. You're comparing it to a memory, your memory of what those colors of the squares looked like. How does that memory, where is that memory? What is being remembered, right? And so it turns out that there's communication between the visual parts of the brain and the prefrontal cortex. And the prefrontal cortex essentially stimulates the visual cortex to imagine what those squares looked like. And people have shown over and over again that if I tap my finger, I can very clearly see visual activity in the finger part of my motor cortex. If I imagine tapping my finger, I also see activity in that same part of the brain. If you put people who have schizophrenia and experience auditory hallucinations in the scanner, every time they hallucinate a sound, pretty much, you can see activity in the auditory parts of their brain. Right? So our brains actually self-stimulate. And so when I'm remembering a visual stimulus, like 3 squares, my prefrontal cortex is actually stimulating the visual cortex as though it's seeing those 3 squares. And that is what you're comparing the information to. So when I get that second set of 3 squares, I'm comparing it to the self-stimulated memory of those 3 squares. And if I have a brain lesion to this side of the brain, then I can't do that anymore. Okay, so how does the front communicate with the back? What is being communicated? So this is the first EEG. This is Hans Berger. He discovered EEG in the early 1900s. He discovered basically that the brain, you can pick up EEG activity from the brain on the scalp in the late 1920s. And this here is activity in the visual cortex that he's recording. And this is a timing signal. So that's a 10 hertz sine wave, meaning it does 1 full cycle or 10 full cycles, I should say, every second. And the visual cortex oscillates at 10 hertz very strongly. This is the oscillations part of my talk. I think this is very critical to how the brain actually communicates. So if I show you an EEG signal that looks very complicated, it may not actually be that complicated. So this right here, this complicated looking noisy mess, which is, let's say, brain activity, which is the electrical activity from a part of a brain, this is actually just a bunch of different sine waves added up, right? So think about, I don't know if anybody ever has much experience in radio, but if you just try and pick up a radio signal from anywhere, really, you hear a lot of static. There's a lot of noise. But if you tune into a specific frequency, suddenly you can hear very clearly a signal in that noise, right? If you block out all of the other frequencies and tune in just one, suddenly you have a very clear signal. And I think this is analogous to how the brain works. So different parts of the brain oscillate at different frequencies. So the visual parts of the brain tend to oscillate around 10 hertz. And the language and memory parts of the brain tend to oscillate around 4 hertz or 8 hertz. And so I think that the brain communicates to itself using these different kinds of oscillations. And I'll show you a little bit of evidence that why we think that might be the case. Oh, that's not necessary. So unfortunately, the signal that we get from out here is not very clear. In neuroscience, working with people, there's always a trade-off between how much information you can get per second from the brain or how much information you can get for which parts of the brain. So let me try and rephrase that. So if you see a study that somebody's published using brain scanning, and you see those nice pictures where they've got blobs of active parts of the brain, that's done using what's called functional MRI, functional magnetic resonance imaging. And that is great for figuring out where in the brain is active when you're doing some sort of task. You have really good what we call spatial resolution. You can get 1 millimeter cube or even less, which is a very small region. Unfortunately, you can only take a picture once every 1 to 2 seconds. A lot happens in the brain in 2 seconds. A lot, trust me. So there's a trade-off. You have great spatial resolution. You know where activity is happening, but you don't know when, because you have bad temporal resolution, we say. Now in EEG, if I put electrodes on the surface of your brain, I have fantastic temporal resolution. I can figure out what's happening once every 1/100th of a second, or actually once every 10,000th of a second even, in your brain. But I have no idea where activity is coming from, because the skull, which sits between your brain and my little recording thing, basically acts like a filter. So imagine if there's like 2 speakers on the other side of this wall, right? And I can hear sort of a right? But it's really smoothed out. It's really unclear. That's kind of the same idea. You've got billions of neurons in the brain firing, and they're all getting smoothed out by the skull, and suddenly I don't know where anything is coming from, right? So if you ask me if there were, let's say, 5 speakers on the other side of the wall, and you ask me, where are those 5 speakers? I would say, I don't know. There's no way for me to know, actually, where those 5 speakers are located, because all I hear coming off the wall is one solid right? So there's always this trade-off. Now, we can get around that if we don't have to have the skull in the way. And so this is a photo from classic, really the big changing moment in the history of neurosurgery, Wilder Penfield, who is this Canadian neurosurgeon in actually Montreal. And he started doing neurosurgery for people who had epilepsy and brain tumors. And what he would do is the people would be put under general anesthetic, so they would just be knocked out, right? And then he'd remove a big piece of their skull and basically open up so that he could have a clear shot of their brain and then wake them back up. So you're now awake with your brain exposed in the operating room. And of course, you're still anesthetized around here, so you can't feel it. Obviously, this is very uncomfortable for people. But what he was doing, the reason that he woke them back up, is because he wanted to make sure that, let's say you had epilepsy right here. This is like the cause of your seizures. What he wants to do is remove that part of your brain, because if you remove the part of the brain that's causing all of your seizures, you're very likely to be cured of epilepsy. The problem is, even though you look at a textbook and you say, this is where movement happens, this is the motor cortex, and this is where language happens, this is the language cortex. In reality, there's a lot of inter-individual variability, meaning your language parts of your brain are not the same as language parts of my brain. They might be several centimeters apart. And all the brains look different. Not everybody's brain is the same shape, right? There's different shaped, it's really messy. And so in order to know that if the epilepsy is coming here, I need to know that that's not your language area, because I don't want you to wake up and not be able to speak. So I want to be as careful as possible. I need to map out exactly the extent of your language cortex. And so in order to do that, unfortunately, you have to have the person awake. And then you take a little stimulating electrode, and they're lying there and they're reading something, like Jack and Jill went, and if he stimulates, he goes zap, and suddenly they go z, z, z. Okay? Then his little assistant comes over and puts a little piece of paper that says piece of plastic that has the number 11 on it, and they've got a clipboard, and they write area 14, speech arrest, meaning that the stimulating there caused the person not to be able to speak. Mind you, this is 60 years ago. This is a picture I took in the operating room last year. Exact same deal. We still do this today. So part of the research that we're doing, my colleagues and I, neurosurgeons at UCSF, Johns Hopkins, and Stanford, as well as researchers here at Berkeley and UCSF, myself included, we're trying to see if we can actually do this better. But I'm not really going to get into the details. But I do want you to notice, still got the little pieces of paper. There's still a person standing there with a clipboard writing things down while the person's brain gets stimulated. And that's actually a video I'm going to play really quickly. You can hear the computerized voice. So the person is actually, what they're doing is you hear the word chair and you're supposed to generate, you hear a noun, you're supposed to generate a verb. So if I give you the noun, chair, you might say a verb. Perfect, okay. That's the task, that's what people are doing. But they're having brain surgery at the time that they're doing it. So you pointed out that you could see the brain moving, right? Does anybody have an idea as to why that hasn't heard me do this before? Blood flow, somebody said blood flow. So the brain is 3% of the overall body's mass. It weighs about 3 kilograms. So it's somewhere between 1 to 3% of your overall weight. But it gets 20% of the blood flow from the heart. So much blood actually with every heartbeat that it actually goes poof, poof, poof. It's like blowing up a balloon, right? So it's very metabolically expensive, we call it. It takes a lot of energy to keep the brain going. Right? 20% of the energy, effectively, 20% of the oxygen and glucose that you take in is essentially going to your brain for this 3% body mass. So why do we do this? Well, like I said, the signal's better. If you don't have the skull in the way, you get a much better EEG recording. So this is actually a study I did at SF General when I was a grad student. This is a group of people who actually had a large part of their skull removed for 6 months. Why would you have a large part of your skull removed for 6 months? I've asked the surgeons to do this to me. They won't do it. Apparently, you can't volunteer for this. No, so seriously, though, this is, I don't know if anybody's ever been to San Francisco General Hospital. Has anybody been there? Can you tell me in 1 or 2 words, if you had to describe San Francisco General Hospital to somebody, what would you say? It's a madhouse. San Francisco General is a madhouse. And they get some of the more interesting patients there. For example, this is a CAT scan of one of the subjects that was in my study. And do you see these little things that look like they're sort of in the middle here? That's pieces of the guy's skull that they didn't want to remove because it's very dangerous to remove things from the middle of the brain. So, okay, this guy got shot twice and walked into the ER in the head and walked into the ER under his own power. Like he walked into the ER and said, I've been shot. And the gunshot had pushed some of his skull into the center of his brain, and the surgeons just leave it there. because you do way more damage trying to go in and take it out than you do just leaving it in there. I had a friend who got shot by a shotgun, right? How many people can say that? Luckily, it wasn't me. And that's a weird thing to say, but he got shot by a shotgun from very far away. And the whole back part of his back and part of his arm was filled with buckshot. So like little, metallic pellets. And they just left him in there because they go in hot. They go in sterilized. And so it's much better to leave 10 or 20 pieces of sterile metal in you than risking infection by going in and taking them all out, right? Because if you take them out, then you have a big gaping wound, and it's more dangerous. Kind of similar here. It's just better to leave it in there. Anyway, so why do they do this surgery where they remove a big piece of the skull for 6 months? Well, if you get hit really hard in the arm or leg or something like that, you bruise, right? So the tissue swells up, you get a lot of blood that goes to that area, and what you call edema, which is sort of the tissues break open and there's some watery, salty solution that gathers in the area around the wound, that causes it to swell. Same thing happens sort of in the brain, where the brain will also swell, except now your brain is stuck inside the skull. Your brain has nowhere to swell to. It can swell two places. One, into the skull. And a lot of the arteries that carry oxygen and glucose to your brain are sitting on the outside of the brain. And so if it presses up into the skull, those get cut off. And when you have a lack of blood supply to the brain, the tissues start to die, and that's what a stroke is. That's no good. Or it could swell down. You have a hole at the base of your skull, which is where your spinal column comes through, right? Your spinal cord, and your brainstem. And if the brain swells down onto that area, it presses onto the brainstem, and your brainstem is very, very important for things like maintaining breathing and a heart rate. And so if that gets pressed on, you lose the ability to do things like breathing and maintain a heart rate, which is also very bad. And so these people would come in with this kind of phenomenon, this swelling, and for decades they were written off. There's like nothing we can do. And the doctors would usually let them die because it's just, there's nothing you can do. It happens too quickly. And the surgeon that I worked with, Jeff Manley at San Francisco General, and some people that he had worked with earlier said, screw that. Let's give the brain a place to swell to. And so brings them in really quickly and just pops out a big piece of their skull. And the brain swells out that way now. And everything is okay. But it takes months for the brain swelling to go down. Usually if the skull is not damaged from like a gunshot, people want their skull back after that 6 months. And so then they can actually put the skull back in place. But for 6 months, those people don't have a skull. It's just brain, and then they put the skin back over for 6 months. So this is my favorite part. The skull is living tissue. And if you just leave a skull sitting on a table for 6 months, it's going to essentially rot, right? It's going to die. You don't want that. That's bad. So how do you keep the skull alive? Well, the surgeon cuts open your abdomen and sticks it up inside and sews you back up. So your skull is being kept alive, basically being bathed in the body's own fluids. There's a woman, I didn't include the picture. I forgot to include it. There's a woman who was the Idaho beauty queen, and she tripped in the forest or something, like some ridiculously minor incident, but just fell just the right way or whatever it was, and basically needed to have this surgery. And they published all of her pictures. She agreed to have all of these pictures published, including a picture of her stomach with her skull embedded inside. So if I can show people afterward. It's pretty amazing. And people that were, just a few decades ago, 20 years ago, 10 years ago, were written off as dead, have an amazing recovery right now. And during that 6 months, when they were bored without their skull, I would bring them into the lab and say, hey, could we put some EEG electrodes on your head there? And basically the whole point of the study was to show that you get a better signal if the skull's not in the way than on the side of the part that does have skull. But you can do some amazing things with it. So that study was actually showing that I can predict when a person made a movement with their hand better because I have better signal on the side without the skull than the side with. And my colleague did the same kind of thing, recording directly from the surface of the brain. So what you're actually going to see right now is people are sitting there, and they've got a pen, basically, and their job is to move to different yellow circles. You make these different movements. And I think it's the red dot. Here, let me play it really quickly. Yeah. The red dot is the actual movement being made by the person, and the green dot is the movement that our computer algorithms think the person is making based upon the electrical activity in their brain. So you can see it's noisier, right? It jumps around a little bit more, but I have to say we're pretty good at this, right? That's pretty cool. We can actually predict where people are moving. There's a lag of maybe about a 20th of a second, just based on the pattern of electrical activity coming off of their motor cortex. And there's actually a guy at UC Berkeley, Jose Carmena, who does this amazingly well, and he's now hooking up robotic arms to these softwares so that you can move robotic arms using this. And this has actually been extended by Bob Knight, who is my PhD advisor, and Brian Paisley, who's a postdoc in his lab. You could do the same thing by recording off the auditory parts of the brain, and based upon the pattern of electrical activity from the audio parts of the brain, you can reconstruct what people were hearing. And Jack Gallant at UC Berkeley is doing this in the visual cortex, while people are getting fMRI brain scans, and you can reconstruct what people were probably seeing based upon the pattern of electrical activity in their visual cortex. So it's pretty cool. This is an example of, actually this is from an animal. They're recording from the visual parts of the animal's brain. And on, oh, that's the wrong citation. That's not right. That's not right. Ignore that in the bottom left. This is from Yang Dan at Berkeley. On the left is, or on the right is the video that the animal was seeing. And on the left is what we think, or they think, I should say, because I wasn't part of this. They thought the animal was seeing based upon the pattern of electrical activity in the visual parts of the animal's brain. So mostly gray. And then something comes into the frame. And so it's not perfect. It's really much noisier, right? But it's pretty good, right? If you were blind and you could suddenly go from being blind to seeing that, you would probably choose that, right? It's really fuzzy, really blurry, but not too bad. And that, by the way, was about 8 years ago. So we've gotten a little bit better since then. This is actually from my friend, the wrong citation in the bottom left again. I added these videos really last minute, actually like 5 minutes before they started, because I thought these were cooler than talking about my own research. So this is actually my buddy Yosef Parvizi. He's a epileptologist at Stanford, and he and I have written a couple of papers together. And he just published this cool paper recently, like last month, trying to do causal neuroscience and show that this one area of the brain is responsible for recognizing faces. We've known for a long time it's probably important for recognizing faces because people that have damage to that part of the brain have what's called prosopagnosia, which means you can't recognize people from their faces. It's actually surprisingly common, and people have it to varying degrees. You literally cannot tell one face from another, which indicates that there's a very specialized part of our brain for face processing. But he went a step further and showed that you can actually manipulate people's ability to recognize faces by working with these people who have these electrodes implanted in their brains. So mind you, these people are undergoing brain surgery, right? And they've got these electrodes implanted in their brains for about a week. And they're really, I promise you, really bored sitting in this ICU. They've got these electrodes in their head, but they can't go anywhere because they're being monitored 24/7. And when we come in and say, hey, look, we're trying to figure out this or that, they're usually really happy to be part of the study because they're pretty darn bored. And you'll see this guy. So this guy has got electrodes in this part of his brain called the fusiform face area that we know is important for processing faces. My colleague Parvizi stimulates this part of the brain, and you'll see what happens. So he does one set of sham stimulation, which is he tells the guy he's turning the stimulator on, but he doesn't, and then one real stimulation.
When I do this, all right? 1, 2, 3.
So he pretended to turn the stimulator on, and the guy says nothing happens.
When we do 1 more time,
Now it's for real.
1, 2, 3.
You just turn into somebody else. Your face is amorphous. Your nose got saggy to the left. You almost look like somebody I'd seen before, somebody different. That was a trick.
So, boom, boom, stimulate for just like a tenth of a second. And the guy's perception of his face went from normal, 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 and then back, right? So again, that's very good causal evidence that this part of the brain is important, because if you add noise to it by stimulating it, it messes up your processing. So this idea of adding noise is, I think, a very important tool to understanding how the brain works. If you can add noise when you're supposed to be remembering something just on that trial and impair performance, and not add noise but pretend like you are and performance is okay, then you can start really also getting at ways of testing the causal relationships between brain and cognition without having to have somebody that has a brain lesion or something like that, right? And so we can do that kind of stimulating, but non-invasively. We call it non-invasive brain stimulation, using a variety of different methods. And we can interrupt communication between brain regions. How do we index communication, though? So one way is by coherence. So remember I talked about these brain oscillations. You have brain waves. So one way that we assess whether or not two brain regions are communicating at any one given time point is if they're oscillating together. If you have two brain regions that are moving activity in a similar pattern, that's statistically very unlikely to happen by chance. That probably means that those two brain regions are communicating. And to give you an idea of what this actually means, this is our little toy model of what coherence is. So these three brain regions are going to become coherent. They start out of sync. They're incoherent. They're not communicating. But then, of course, they can become coherent if they are communicating. Right? And so that's a visual representation of what we mean when we're talking about coherence. And that's it. That's really it. Like, are they waving at the same rate or not? That's essentially it. And so if two brain regions are oscillating together, then we think that's a really good, strong indication that they're communicating. And so in my research, I have tried to show what is being communicated. And I'm not really going to get into the details, but this is a paper that we have under review right now that I'm pretty excited about, working with 4 people who have these electrodes implanted in their brains. And it's sort of this FM radio hypothesis, showing some evidence that they may be the case, that different brain regions are tuning into different frequencies. And so we can start manipulating brain activity a little bit using non-invasive brain stimulation. And that is something we refer to, like I was mentioning earlier, plasticity. We can modulate activity. And the capacity of the brain for changing itself is pretty amazing. I'm going to show you, this is from a paper in 2007 of a little girl who was born with that much brain, 2 days old. She had hydrocephaly. And huge amounts of her brain were missing. All this black here should be brain, but that's just space. And at 20 months, this is what it looks like. And by 34 months, this is her walking and pushing around a little cart. So, of course, your mileage may vary. If I take out that much of your brain, you're not going to be okay. But the fact that it happened to her at 2 months or 2 days old, or even in utero, was probably better for her. There's actually cases of babies being born essentially with like seizure or something like that, and the doctors will actually perform what they call a hemispherectomy, which is they remove half of the brain, one whole hemisphere. And kids that have that done early enough have normal IQs and everything, because it just sort of kind of regrows, and they reshape and they readapt. Patients who are blind at birth, the visual parts of their brain readapt. They get used for other stuff. They start picking up sensory, motor, or like touch sensations, I should say. So it's pretty remarkable the degree to which plasticity happens in normal situations. And it happens a lot in normal aging. And it's been shown over and over and over again that if you, as we get older, our performance slows, our reaction times slow down, we can't respond to things as quickly, and there's a shift in the way the brain seems to process information. Instead of processing it, like if I present you a visual memory stimulus, like I showed you earlier, those 3 squares, a 20-year-old would rely very heavily on the visual parts of their brain to do that task. The sensory parts of their brain process most of the task. the cognitive prefrontal cortex is not very active. It's not doing a whole lot. It's not needed until it becomes very, very hard. In older adults, that activity shifts to the front of the brain. And so instead of relying on the perceptual parts of the brain, people start relying more on the cognitive parts of the brain. They work harder to fill in that missing information. And there's a hypothesis that's been long-standing for 40 years called the neural noise hypothesis, which is why people think that this is the case. And the idea is that as we get older, the sensory parts of our brain that should be processing information very clearly just get noisier. Something is breaking down as we age. And this is about a 40-year-old hypothesis that has no experimental evidence whatsoever. Yeah, I should say until now. So this is an example of how our behavior changes with age. So young adults, remembering 1, 2, and 3 items, respond on average at about 480 milliseconds. It takes them about half a second to tell me if it's same or different. And it increases slightly as it gets harder. Old adults have that same pattern where it increases slightly as it gets harder, but they're about 150 milliseconds slower. Not a huge deal, but 150 milliseconds can be a long time in certain situations like driving a car, right? So I looked at 16 of our patients that had these electrodes implanted in their brains, aged 12 to 53 years old. And I said, is there more neural noise in the auditory parts of their brain? And the trick was trying to figure out how to define neural noise. I'm not really going to get into that. There's a reason why nobody's been able to do it before. And this is a paper that we're submitting hopefully within the next couple of weeks. But the long and short of it is we show that neural noise increases as a function of age. And if you're interested in the details of how I calculate that, because I'm running out of time, I'll gladly talk to you about it after this. And so this got us thinking, Adam Gasly and I, okay, 150 milliseconds is a long time. Neurons are noisier, and we think physiologically we understand why that's happening. And there should be consequences of this. How can we look at that? And so there's a company in San Francisco called Lumosity. Does anybody know who they are? They do online brain training games. And their online brain training games are the boring cognitive tasks that we have our undergraduates do, just made slightly more into a game. So this is called a flanker task. It's a very common task that's been done in psychology and neuroscience for about 35 years or so. And they just made a game out of it because they just made them into birds instead of arrows. And they give it a nice background. And your job in this task is to tell me which direction is the central bird facing. That's it. You press up, down, left, or right. People are very good at this. If the 4 surrounding or flanking birds are all moving in the same direction as a central bird, you respond more quickly and more accurately. And if that center bird is facing in a different direction from the flanking birds, you respond less accurately and more slowly. And the reaction time difference between these two is the interference effect. How distractible are you by flanking information? You don't need to pay any attention to these birds. You're supposed to not pay attention to those birds, but you can't help it. Your brain still processes that information, and we can get an index of that. And so it's essentially how distractible are you by distracting information. And they have 300,000 people that have done this. So instead of looking at 15 undergraduates in the lab, they now have 300,000 people from around the world. Suddenly, we have a huge amount of data that we have access to. And this is people's response times as a function of age for all. This is actually 30,000 people. So 30,000 of the people that they have data on did this game at least 25 times. And so you can see 20-year-olds on average take about 700 milliseconds to respond. And about 1% of 70-year-olds can respond that quickly. And in fact, on average, we get about 4 milliseconds slower per year of age. And by the time we're 70, we start getting about 15 milliseconds slower per year. Now again, 4 milliseconds is not a lot. That's 4 1,000ths of a second, right? But across 40 years, that suddenly becomes a long time, right? So here you can see the average 19-year-old, the average 20-year-old, the average 21-year-old, 22-year-old, and so on and so on and so on, until by the time you're 69 and 70, you are responding half as quickly. I'm already responding about 25% as fast as I was when I first started neuroscience. And so, what do we do with this much data? Well, I pulled in data from each US state and said, what are the rates of motor vehicle fatalities in those states? What are the rates of ADHD? What are the IQs of that state and the number of high school graduates, percent of high school graduates, I should say? How many internet users, autism rates? And it turns out that your distractibility on that task is correlated with the percent of motor vehicle fatalities by population in each state. So these are the different states. California, subjects on average, not too great. They're pretty distractible. Alaska, surprisingly, okay, actually. Very not distractible. Alaskans are very resilient on average. Mississippi has a huge number of motor vehicle fatalities. So does Wyoming. Kentucky is very distractible, apparently. Now, mind you, correlation here, again, does not equal causation, right? This is an exploratory thing. It's the question of, okay, given the amount of data that we have now, can we learn something new? IQ correlates with working memory. This is sort of our check to make sure that the data we're getting is very good. This is very well known. People with higher IQs have better working memory. States with higher IQs also have people with better working memory. That should not be a surprise. So this is kind of a little bit of a sanity check to make sure that the data might be good. And North Dakota, very, very good working memory and very high IQ apparently. It's kind of interesting to look at the states. I'm not going to get into anything political here, but there is some commentary that one could make, I'm sure. So what, right? What about this? And so I'm going to close out with some of the future directions that I'm hoping to do. Like I said, very few 70-year-olds respond as quickly as the 20-year-olds. But there are some who do. Why? Right? If I can reach out to these people and bring them now into our lab and say, what is it about their brains that is different? What allows them at 70 years old to do just as well as your average 20-year-old, right? And you can actually question them. You can get information about their dietary health and their lifestyle choices and whatever, right? We have large-scale behavioral, we could never ever do this in the lab, right? This is 100 people out of 300,000, right? But we have enough data now that we can start maybe reaching out to those people and bringing them into the lab and actually seeing. And so we've actually started the process of making games out of our cognitive tasks. So do you guys know what the Xbox Kinect is? Right? So we took one of our attention tasks and we made a game out of it. And I'm not very good at it. So it tells me where I'm supposed to pay attention, and if it's a green thing, you reach out to it and you touch it. And we're publishing a paper right now showing that using the Xbox Kinect, we also did the same task on an iPad. You can actually get research quality data. We can get the same kinds of behavioral effects that we get in a computerized lab test as we do on these systems because they're good enough now that the data quality, we can get reaction time information very well. And so our hope is to actually just start releasing these things for free. Like I'm not trying to sell anything. I don't have any monetary interest in this. Our hope is to just release these things for free and get people give us permission to record the data and then potentially contact them in the future to bring them into our lab to actually do some brain-based studies, right? So there's this sort of famous article in Wired from a couple of years ago saying that data is the end of the scientific method. We don't need hypotheses anymore because now we have data. Data can tell us everything, which is great, except wrong. There's a fantastic, do you guys know Radiolab? It's a WNYC NPR show. And they had this fantastic article, story. And it was an inspiration for that data mining study that I showed you at the very beginning, where he took the text of 3 million neuroscience articles. And what these scientists did was they hooked up a video camera to watch a double pendulum swing. I don't know if you've seen a double pendulum. A pendulum just goes like this, right? A double pendulum is another one on top of that, like underneath that. And so this one swings and this one. And it's extremely chaotic looking. If I have you try and predict when that second pendulum at the bottom loops through the first one, you cannot do it. It doesn't behave in any way that looks like it should. But of course, the rules that govern it are basic Newtonian mechanics. F equals ma. That's it, right? So it looks very chaotic, but there's a very simple rule governing the motion of that system. And these guys set up a video camera and recorded the motion of the system, and basically just had a computer say, try and figure out some equation that fits the behavior of that. And in 2 minutes, their computer was able to deduce F equals ma of 2 minutes of observing a double pendulum swinging. What took humanity 100,000 years until we got to Newton to do, a computer did in 2 minutes, right? It was great. It was a really cool paper, and it was a huge breakthrough. And these biologists said, oh my God, this is a problem we've been having forever, which is trying to understand how all these different parts of cells interact. Cells are extremely chaotic looking systems. Can we just set up this video camera and record the cell and see how all these proteins are interacting in the cell and come up with a set of equations that define how these proteins interact? And so these guys set up their camera and they set up their equipment and their machine learning algorithms, and within a few minutes, they deduced out these equations that perfectly predicted the way that these proteins in the cell were interacting. And they had all these variables, but it works. It works extremely well. And everybody said, we don't know what any of these variables mean. So they have an equation that matches the system perfectly, but they don't know what any of it means, right? So yes, data is very good for us, but sometimes we need somebody to actually interpret it and do something with it. And so I believe in the power of data to inform us about something new, but then we can bring those people in and actually start interrogating them from a hypothesis-based direction to say what is different. So we use the data to inform the science, not the other way around. Anyway, so we've got these noisy systems, right? 100 billion of these things. Actually, that's been revised now. It's probably more like 85 billion of these things in our brain. And there's 7 or 8 billion of us running around trying to interact with each other. And at the end of the day, we still have this. All right, thanks everybody.